All right, hello everybody. I'm Frank Slack. I'm the director of the HIRM at Beth Israel Deaconess Medical Center, and today it's a pleasure to introduce one of our own, Dr. Todd Woolf, who's actually the director of the Tech Ventures office here at the IDMC, who's going to be our seminar speaker for today. He actually has quite an illustrious science background. He's not just a businessman or sort of a techie. Actually, Todd did his bachelor's of science in cellular molecular biology at University of Michigan, and then did a short stint in Leroy Hood's lab at Caltech, looking at solid phase isolation of genomic DNA and some of the things that ended up becoming very important for DNA sequencing. Then went and did his PhD here at Harvard in Professor Doug Melton's lab in biochemistry and molecular biology. And from there, he moved into industry and has worked at a number of the real sort of innovative RNA companies and sort of oligonucleotide companies along the way, like Ribozyme, which became Sirna, that was bought by Merck. And he worked in Ontogeny and Sequitur, and then started RXI, and then started Etigen, Epifini, and then ultimately moved out of that space and became a licensing officer, first at MIT, and now he's the director of Tech Ventures here at the IDMC. It's wonderful to have Todd with us today. The title of his talk is going to be Taming the Helix. So Todd, do you want to share your slides and take it away? Thanks.
Well, thank you, Frank, for having me to talk here. And today I'm going to talk about the first 40 years of RNA therapeutics. And it will go over work from my work with my groups over a number of years, and also selected work from other people in the field that followed up and extended this work. The first slide shows, one second, I'm trying to get rid of this thing. All right. We're going to talk about RNA therapeutics in three different categories: silencing mRNA, downregulating the expression of RNAs, therapeutic editing of both RNA and DNA, and chemically modified mRNA therapeutics and vaccines. In terms of silencing, I'm going to start where it really began, with antisense oligonucleotides that can cleave the target RNA. But first, I'm going to talk about the chemistries involved in the technology. So the baseline chemistry is unmodified DNA. And the problem with that, I mean, the field would have been done if that was stable, but it has low endo and exonuclease stability, so we had to use chemistries to stabilize that. And it also has a property that it's a substrate for RNase H, meaning that if this binds to a target RNA, RNase H will come along in the cell and cut the target RNA. Early and most important chemistries in the field is phosphorothioate to stabilize the backbone. And this is shown schematically in my slides as red. This is when the non-bridging oxygen is a sulfur. And these have a number of unusual properties, and a lot of it was just accidental. I think they were meant to stabilize the DNA, but they also led to some toxicity and lower affinity. But magically, they're still a substrate for RNase H, one of the few chemistries in all the years of oligotherapeutics that retains the ability to activate RNase H when bound to RNA. They happen to be sticky, binding to proteins, which allows them to have a good biodistribution and get taken up into cells. And they have high stability, about 1 to 3 days, not super high, but good enough. Another chemistry is 2' modifications, which I also throw in LNAs. And the 2' position is, of course, the position that makes it RNA or DNA. And these have another number of great properties. They can be very high affinity. They're varied in nuclease stability, and they also have a lower immune stimulation than unmodified DNA. So that makes them useful. Another area of modification is the nucleotide bases, and these can increase lower immune stimulation if you modify them, and increase hybrid affinity. An exotic form of modification is neutral modified backbones, phosphoramidates, PNAs, morpholinos, methylphosphonates, and these have are so foreign that they're not recognized by the cellular machinery at all. but that makes them extremely stable. They're not a substrate for RNase H, and they have very low toxicity. And lastly, but very importantly, is just linkers linked on the ends of the oligonucleotides, shown as a purple triangle here, that can act as delivery ligands or exonuclease blockers. And believe it or not, every one of these chemistries have been used at some time or another in important oligotherapeutic candidates.
The field of antisense therapeutics began in Massachusetts back in 1978 when Paul Zemechnik, who also discovered transfer RNA, did the first antisense experiments. And in fact, those experiments actually used chemically modified oligonucleotides. They had N-blocking groups. An interesting note is when I asked Paul about these early experiments and various controls, he said it was very difficult to do a lot of controls because it took them 1 year to make the oligonucleotides, chemically, manually make them. So Paul received the Lifetime Achievement Award for this work and other work. And that was the beginning of the field. Now, these antisense oligonucleotides, at least the first ones, were substrates for RNase H, which meant the oligonucleotide would bind to the RNA, and then the endogenous RNase H enzyme, which recognizes DNA and RNA hybrids, would cut the RNA, destroying it. And usually the ends would also be degraded eventually in the cell.
So right off early on, Charles Jennings in our lab, when I was in Doug Melton's lab, saw a problem here with antisense. People would show these antisense oligos of 10 to 25 long and multiply how many combinations there were and say they'd be incredibly specific. But Charles noted that if you had a small region that perfect identity in a row, that would make them much less specific if that could activate RNase H. Or if you had mismatches that were active, it would be radically less specific than originally envisioned. So I did some experiments and we actually saw that occurring. Here we were targeting a gene in a Xenopus egg.
Anastasia doesn't have a last name.
At the target site. And in other site within the same RNA where 10 out of 13 happened to be identical, by chance, we saw partial cleavage there too. And Anastasia followed up with this in that she saw the same sort of things happening, Anastasia Horva, years later with RNAi. And of course, the same thing is a concern with CRISPR editing. But in each of those cases, through sequence design and chemistry, increase the specificity. Next slide. So the first generation oligos were 100% phosphorothioate DNA for the most part that were used in the clinic. And these required fairly high doses. And unfortunately, although they had some activity, there was only one drug approved out of many trials. All the rest failed. These phosphorothioates were designed by, the chemistry was made by Cy Stein and Jack Cohn at the NIH. Fortunately, these chemistries were found to still be very important if used in combination with other chemistries. And that's what you'll see a lot of my talk in many instances. There's phosphorothioates used either on the ends or in combination with other chemistries, and they turn out to be quite critical. So while uniform modified oligos weren't working well, Joe Walder at IDT and University of Iowa proposed making these gapmer designs with, in this case, phosphoramidates on each end with a gap in the middle that was DNA that could activate RNase H. And in the Xenopus eggs, we injected these, and here is a control gene, RNA fibronectin. There's the target control oligo, and you see this just wipes out the targeted mRNA on the northern blot. So this was very good as a research tool, but it wasn't stable enough for use in therapeutics or mammalian cells. At Ribozyme Pharmaceuticals, which became Cerna, my team was working on the rules for stabilizing ribozymes. And we found that if you had unmodified RNA in the middle of an oligo, it wasn't so stable. But if you substitute that with 2'-methyl, even though there wasn't phosphorothioates in these linkages, it was highly stable, highly potent. If you had 2'-O-methyl on the ends, it wasn't very stable or potent. But if you put 2'-O-methyl and phosphorothioates on the ends, you had high potency and high stability. So this sort of was working out the rules for how you could make stable oligos and perhaps reduce the amount of phosphorothioates as seen here in a molecule to reduce the toxicity. We took that information at Sequitur, a company I founded, and we made these antisense oligo designs where you had an N-blocking group, then you had 2'-O-methyls without phosphorothioate to reduce the toxicity and to increase the hybrid affinity. And then at the end, you used phosphorothioate DNA, which is a nuclease blocking group, and also activated RNase H. So these were very good research tools, and they were used to validate the beta secretase by Amgen and Pharmacia, who were our clients at that time in 1999. They inhibited the target RNA compared to control. There's 3 different antisense oligos here. Each of the antisense oligos worked compared to control. And that also inhibited APP processing, which was the proof that this was the beta secretase. But those oligos weren't optimized for therapeutics, and therapeutic application of antisense cleavers was quite a tour de force that occurred over many years. Required technologies from the NIH, IDT, IDERA, Sudhir Agarwal's technology was licensed in, and work done by Centeris. And a leader in the field, of course, is Ionis. They started out with these all phosphorothioates that were required high doses. Then they used the Gapmer design with 2'-methoxyethoxys that had higher affinity and required about 10-fold less dose. Then the LNAs came along, which they call sets, their version of them, which are even higher affinity with the GAP, and they were down to 15 milligram doses. And then remarkably, with their current leads, a GalNAc conjugate with the linked RNA on the ends and phosphorothioate, basically everything in the kitchen sink, they're down to a remarkable dose of about 1.5 milligrams per week for ID50 in humans. So it took really everything to get these things to work, but now they work quite well. So in the midst of the RNA doing antisense, RNAi came along. Craig Mello and Andy Fire discovered these compounds that originally were unmodified, just 2 strands of RNA that were quite good at inhibiting targets. And they were thought to have all these great properties, and they did in cell culture, but they required a lot of chemistry and modifications to get them to work as a therapeutic. All the unmodified siRNA clinical trials failed. So it really took the same sort of thing, chemistry, to get it working. So I'll talk about that. At Sequitur, we used what we called stealth RNAi, and that had some 2'-O-methyls linkages on the ends, which increased this nuclease stability, shown here. Here's without the 2'-O-methyl linkages compared to the stealth in serum. And it reduced the toxicity by reducing immune stimulation. Here's immune stimulation with siRNA, and with the modified stealth, there was less stimulation of these cytokines. So these were quite useful as a research reagent, not stabilized enough to be used as naked RNA for therapeutics, but they've been used in many functional genomic studies and are sold by Thermo Fisher at this point. In terms of therapeutics, I worked with Anastasia Corvo and Dmitry Samarsky and others at RXI Pharmaceuticals, and we developed self-delivering RNAi, which had a lipophilic group, a tail, a single-stranded region that had phosphorothioates, which was helpful for sticking to the cells and pharmacology, and then a double-stranded region, which loaded into and activated the RISC complex. These, when you put into cell culture, get into the cells, and even in animals, they get into the cells and inhibit the target genes. We did a clinical trial with these at RXI Pharmaceuticals, targeting a target in the skin for scarring. And this shows in the animal model, the complementary oligos inhibit it, where the control oligos did not. This was the first delivery conjugate to enter clinical trials, but it was those trials were stopped for commercial reasons in phase 2. So more excitingly was the work done by Alnylam with mono. And they put together, again, using licenses and technologies from a number of groups, they made an incredibly complicated molecule that had a Gale neck on the end, had 2' modifications, some phosphorothioates, modifying every linkage and in some way, every nucleotide in some way to make it stable to enter the endosome. But when they put this all together, they were able to get tremendous inhibition in humans with dosing, and the inhibition even lingered after you stopped dosing for quite some time. And while this one didn't make it through to approval, they've had approvals, and this is now one of their lead chemistries for work in the liver. So these self-delivering technologies are now, of course, following the great results with Alnylam. There's many groups doing this. Dicerna is getting great results. Silence Therapeutics is using it. Adverna, company I work with, is using it as a research tool. A new company, Atalanta Therapeutics, with Anastasia and others, has a very interesting version of self-delivering oligos that they're pursuing. And Arrowhead Pharmaceuticals is also using this, as well as FIO, which is the new name for RXI. So now I want to go from knocking down or silencing mRNAs to therapeutic editing of RNA and DNA. So in 1998, we published this generalized model for therapeutic editing where you could edit mRNA by recognizing the mRNA and then having a modification activity. But recognizing mRNA was pretty easy. You could use an antisense oligo. Now they're also doing this with specific RNA binding proteins. But the trick with using an antisense oligo, this is almost the opposite of antisense. You want to use something like 2'-O-methyl or a chemistry that doesn't activate RNase H, so you don't cut the target that you're trying to fix. Then you need the modification activity, and an interesting one is nucleobase modifications. And deamination is one of those. I'll show you an example of that. Exon skipping is another way to repair an RNA. I don't know if you'd call this editing, but it's in the same general class. And transplicing developed by Solinger. In terms of targeting DNA, you have to recognize the DNA, which is harder because if you either have to get a binding protein or a strand invading oligo or triplex oligo, and then modification activity, which again could be a deaminator or base modifying activity. You can cleave and do ligation, as we described in the 1998 paper. Recombination, as Kamek described, or mismatch repair, are all mechanisms how this could edit your target DNA. So let's look at RNA first, because that's where the first work was done. In 1995, we were using this model in Xenopus eggs to prove out the idea of therapeutic editing. And we chose to do RNA basically because we figured a trick on how to do that easily, because this was just a side project and we didn't have a lot of money to spend on it. So the trick was based on some work in the area of natural RNA editing, where it was known that if you form a double-stranded RNA duplex in a cell, the adenosines would be modified to inosines. So here we targeted a premature stop codon in muscular dystrophy in a message fused to luciferase, and that UAG was turned to UIG, which is translated as a UGG, which is not a stop codon, but a read-through and loud read-through. So that was the little trick that we used, and we tried it. Jennifer Chase did the actual work in the lab in my group, and remarkably, this was the first experiment. It worked pretty well. We had the control RNAs, which didn't have the mutation, and they gave us this much luciferase, and then a control oligo or the complementary RNA oligo, and we got about 28% read-through using this, this technique. Now, as expected, the change was not just in the targeted stop codon, but it was in other codons in this general region where the duplex formed. And the interesting thing here is that changing an A to a G can fix any of the 3 stop codons, but it can never create a stop codon. So this still allowed for read-through in this example. Of course, we looked at chemical modifications when we did this, but we did some initial work. We found that 50-2-mer worked the best, a 34-mer worked well, but not as well as the 52-mer. And then we put these N-blocking groups on phosphorothioate 2'-O-methyl, and we saw that we got some activity, but it was diminished. And unfortunately, when we made them all 2'-O-methyl, which would have been a great chemistry to use therapeutically because it's nuclease stable, we lost all activity because it didn't presumably interact with the enzymatic machinery to deaminate the target anymore. This work was followed up by Maria Montiel Gonzalez and Rosenthal. In cell culture, they used a scheme where they, again, used oligos to direct a deaminase activity. Here's the positive control. Here's the mutated sequences in cell culture. And they, again, had read-through to GFP in this case. And when they activated the deaminase towards the target, they saw a nice expression of the target gene read-through, and they could sequence it and show that correction. They were from the University of Puerto Rico, although both of them are in the US and in fact in Massachusetts at this point. So currently there's quite a few companies working on this base editing. Edigen, we basically have some IP in the area. Procure, Beam Therapeutics, Locana, a newer company, Coro Bio, another new company, and Shape are all pursuing this for various applications in RNA editing and some even for DNA editing. So going back to the general idea for recognizing the target and then modifying, you can use exon skipping. And I'll talk about that now, because interestingly, that maybe because it's simpler to do in concept and in practice is furthest along in the clinic, actually has approved drugs. And one of the chemistries used in this in approved drugs is morpholinos. They're a neutral backbone that were developed by Jim Semerton, amazingly back in the 1980s. And these have extremely high activity, extraordinary specificity, and are very stable against nucleases. And Rizard-Cole had a strategy to use oligonucleotides to change splice patterns. And in some cases, for some particular genes, you can use that to actually repair a defect or at least ameliorate the effects of a defect. For example, if you have a stop codon and you block the splicing of this exon into the RNA, you will not have that exon, but you can have a read-through and you won't hit the stop codon. And in limited cases, in certain cases, that will lead to a functional protein if the exon is not critically important and can treat the disease. So my interest in morpholinos came from their low toxicity. Here's an experiment also done at Harvard Dental School, actually, by Malcolm Whitman, collaborated with him when I was at Sequitur, and injecting this into embryos as they developed. If you inject water, they're fine. If you inject morpholinos at 200 micromolar, which is a very high concentration in the cell, they're also developed normally. However, if you inject phosphorothioate DNA at a 100-fold lower dose, the embryo is not doing very well at all, basically lyses the whole embryo. So they're very attractive chemistry. And around that time in 2002, believe it or not, antisense was in such disarray that some people didn't even believe it was true. And Rizard-Cole, as I said, was working on splice blocking. And he had a nice model where you would treat a cell with a splice blocking oligo. And if it blocked a splice site, it would correct defect in the RNA of beta thalassemia, but it would correct this stop codon or skip over the stop codon and lead to expression of GFP. And I suggested that he take his model and move it up into a mouse model and express it in every cell in the mouse. So then you could simply treat the mouse with these antisense oligos and look for a gene turning on. And that would be much easier to see in whole body studies, whole animal studies. And it would also, I think, be more, I told him it would be more convincing to see something turn on than turn off. And remarkably, he actually did this. And they saw beautiful expression of genes that occurred when the splice blocking oligo got into cells. It wasn't surprising that it worked in the liver, and he did this with Mano, who was at Ionis at the time. But it was surprising that it worked in the muscle, and that led to him thinking about other targets. And one target was muscular dystrophy, where you could use this splice skipping idea quite dramatically, since there were so many exons that weren't necessary in the very long dystrophin molecule. And he collaborated with Sarepta, and they did some great work where they had a mouse model of muscular dystrophy that if you looked at the normal mouse, they had nice muscle tissue shown here in a stain. If you looked at a MDX mouse model for muscular dystrophy, the muscles didn't form correctly. And if you put one of these methylphosphonate oligos with a conjugate for self-delivery, you got almost uniform good expression and great results, healthy muscle in all the muscles throughout the body. So a very dramatic, you could look at this reference result that really, to me, proved beyond a doubt that antisense was working inside the body, inside animals, so long as you had delivery. Now, you would think they would go to the clinic with this compound. It was so impressive, but the conjugate did cause some toxicity. So the company was more conservative and went with the methylphosphonate without any conjugate. And that did not work as well. If you look at the next slide, you can see the, excuse me one second, you can see the exon skipping only occurred in a couple percentage of the messages. They do have some histology that looks like it's working well, but it is kind of in dispute how well it's working. In any case, they were able to get this approved as the first drug to treat the mechanism of the defect of muscular dystrophy, and they even have another drug in that class that has been improved, and they're working on conjugates that would be less toxic, that might have the excellent delivery and the low toxicity of the morpholino to get even better correction. Very dramatically, Frank Bennett at ISIS, which is now called Ionis, led a group that did splice blocking with a totally different chemistry, all phosphorothioate, all methoxy modified. And these were infused into the CNS and treat spinal muscular atrophy. This has been approved and is sold on the market. And Frank was and his team was awarded the breakthrough prize for this very dramatic work. I must say, it's amazing to think that this could work, that you could infuse these oligos into the central nervous system and get such great results. And it's a really tremendous example of the technology and a great group of scientists' work. So now I'm going to switch to DNA targeting and talk about mismatch repair. So I have this schematic here, and I put in Bob Brown because he talked about this ideas with me back when we were at Genta in the early 1990s. And the idea here is you have a modified oligo that strand invades and hybridizes to one strand of the duplex. And the oligo is a guide. It's actually a donor DNA that has the wild type sequence. So there'd be a mismatch between the oligo and the genomic strand. And endogenous cellular DNA repair machinery could correct that, and that could become a permanent change, a correction in the DNA. Now, the cell could also correct the oligo, sort of correct, and that's not what you want to happen. So that'd be an unproductive correction. And that is one reason why this technique could be less effective or have a lower efficiency. But I'll show you chemistry can address that. So here is some results that we did at Edigen Pharma more recently, several years ago, in cell culture, where we took oligonucleotides without any CRISPR, without any programmable nuclease or nuclease, just oligonucleotides, and they were transfected into cells. One was completely unmodified, and each oligonucleotide was given to the scientist who did this, our COMEX group, blinded in pairs, and one was wild type and one was mutant. So the mutant wouldn't correct it, but it was only one base difference, or very good control. And we tried various chemistries. So the unmodified DNA, as predicted, wouldn't work very well because it's unstable. The one with phosphorothioate on the ends showed editing activity, a low levels, 0.8% for treatment, but very clear over background with the control mutant oligo. And the phosphor, this one here with 2' modifications still was consistent with editing. One with methylphosphonates on the end had editing. And when we put 5-methyl Cs, we actually got slightly enhanced editing. So going back to this idea of targeting the editing to the mutant strand of the genomic DNA, this is a schematic of some great work done by Bertoni and Rondo at Stanford years ago, where they used oligos that had a 5-methyl C, a GC 5-methyl C. And when it hybridized to the mutation, it formed a TG, there was a TG to CG mismatch, or GC mismatch. And that activates a special MBD4 repair machinery in the cell that actually directs the correction to the target strand, to the genomic strand. And that one change, having a 5-methyl C, one 5-methyl C in the molecule in this, for this particular mutation, increased the editing efficiency by about 4-fold to up to 3 to 4% per treatment. We did some more work at EDIT1 where we saw that we could modify the 5' end with 2' modifications and the 3' and still get editing. And I'll show you why we're interested in these things as I go further in the talk. And what's really important to note with this form of editing is that the transfections can be done repetitively, or the treatments with the oligonucleotide can be done with the animal repetitively, because unlike programmable nucleases, you're not getting the indels, you're not destroying some fraction of the chromosome. You're just correcting some fraction, and if you treat again, you'll get more chromosomes corrected, a higher percentage, and it will just keep going up and up. And this is from the literature, data from the literature. So the efficiency is still low, and one way to increase the efficiency is based on using neutral molecules, actually, to act as a sort of chemical CRISPR. We found back in the early 90s that you could form these triplexes with a single strand of nucleic acid and neutral oligos. And these triplexes would block the progression of a polymerase, in this case, reverse transcriptase. So Glaser and his group have been using PNAs that form these triplexes on the DNA in the region of the editing with the other donor oligo. And the combination of those two oligos without any proteins or cleaving molecules can get quite efficient editing. And here's some work from the literature that he did independent of us that shows that if you have a mouse model for beta thalassemia, it obviously has reduced hemoglobin. And if you treat that mouse model with a editing oligo, N-blocked editing donor DNA oligo, and one of these triplex forming oligos that forms a complex near the editing site, and you do 4 treatments, you get about 4% editing. And that 4% editing is enough because these edited cells have a survival advantage to get normal hemoglobin levels for the as long as they measured in that mouse. Many days, many months after treatment with the editing oligos. So that's an exciting area, and they started a company based on this particular technology. So what my interest is going forward is to find a way to make self-delivering editing oligos so we can edit with a single oligonucleotide with no liposomes and use the advantages that they know of ways to deliver oligonucleotides, use the chemistry to direct editing to the target strand to stabilize the oligonucleotide and to reduce immune stimulation. So this is a work in progress, but that's the goal, to create a third generation self-delivering editing oligonucleotide. And these oligo-only approaches have multiple advantages, as shown here: reduced immune stimulation, no DNA cleavage. And if you just look at them, you have an oligo or one or two oligos. It's a much simpler drug to manufacture and deliver than engineered nucleases and CRISPR, albeit less efficient per treatment. And I'll just end editing with an idea I'm throwing out there. I'd love for somebody to try this, but I really don't do work with CRISPR, haven't had work done work with CRISPR. We didn't have a license, so we didn't pursue it. But I think an interesting combination, which is somewhat similar to prime editing, but a little more simple, is to use dead CRISPR and one of these editing oligos as your CRISPR RNA. And you have to replace some of the linkages with DNA in order to activate the endogenous repair machinery. But it's known that at least some of these linkages can be replaced with DNA in the CRISPR RNA. And if enough of them can be replaced either on the ends or, right, in any case, across from the mutation, you might be able to achieve highly efficient editing by this mismatch repair mechanism. Just a proposal there. So I'm going to finish up the last few minutes and talk about chemically modified mRNA therapeutics and vaccines, also known as mod RNA. So we went from silencing to repairing RNA and DNA. Now we're going to talk about expressing proteins with mRNA. So the idea here is very old. Feldner and Malone had tried it early on with cationic lipids. You take messenger RNA, you transfect it into a cell, and you get expression of proteins. So that's mRNA. But what I'm talking about, what we did at Sequitur, was chemically modified mRNA as a therapeutic or vaccine. And there's where we add some of these chemical modifications to try and improve the properties of the mRNA as a therapeutic or vaccine. And the advantages, and this is a slide from back in 1998, just to show you that the advantages remain, that we proposed was that it's gene-free, so there's no integration or long-term effects, which people were shocked at this. Most people said, why would you want to use mRNA? DNA lasts forever. But we were like, well, sometimes you don't want it to last forever, particularly for vaccines. It's also no immunogenic vector proteins, which is a distinct advantage. And what is kind of not generally known at the time is that mRNA can be expressed more readily in primary cells in many cases than DNA, because it doesn't require nuclear delivery and assembly of transcription. You basically just have to get the RNA in the cytoplasm and it expresses. So we worked on this at Sequitur. We got a partnership with Inex, which is now what became Tecmera. Now they have a different name. But this company funded us to do some work, and we filed a patent on basically every way we could imagine to stabilize and improve an mRNA through chemical modification and sequence changes for use as a therapeutic or vaccine. One of the things that we did achieve is to use phosphorothioates and showed that you could have phosphorothioates incorporated into the mRNA and still get translation in this in vitro translation work done by Peggy Taylor and Kristen Wiederholt. And we found, as it predicted, it was quite easy to make this mRNA, and we made about 1 milligram of that and sent it to our partner Tecmera for some mouse experiments. This was early on, so we just sent them unmodified RNA. It had a cap and a poly A tail, but it wasn't especially chemically modified. And shockingly, it worked pretty well. In the first experiment, they injected this naked mRNA into the muscle. I'm not even sure we suggested they do this. It didn't seem like it would work, but they did it. And after a few hours, we saw detectable expression of the luciferase reporter. And if we compared that to DNA, which had a strong promoter, it had done at the same time or in parallel in animals, the expression was actually better, the mRNA. Tecmera went on years later to use their improved delivery technologies to deliver mRNA expressing genes to the liver. and got beautiful uniform expression as shown here with this M-cherry protein reporter in the liver. Unfortunately, many of the protein mRNA therapeutic programs failed due to toxicity due to immune stimulation. So they're able to get expression, but repeated dosing for long-term wasn't compatible because of the side effects due to immune stimulation, despite the fact that the mRNA was somewhat chemically modified. You just couldn't modify it enough to get rid of enough of the immune stimulation. But of course, immune stimulation is fine when you're talking about vaccines. And in fact, 2 of the 5 primary US COVID vaccine programs are using intramuscular injections of chemically modified mRNA. That's the BioNTech Pfizer and the Moderna. And very exciting to say that those, one has been approved in England, I think just the other day, yesterday maybe, and hopefully these will be approved shortly in the United States. So with the last slide, I want to just summarize the thesis of the work is that successful chemistries are necessary for RNA therapeutics to work. And the chemistries that have been successful direct endogenous cellular machineries while imparting nuclease stability, delivery, and or reducing toxicity of the molecules. And you've seen examples in the form of silencing by antisense or splice skipping, which has to interact with RNase H or the spliceosome. You saw examples of RNAi, which have to interact with the RISC complex, yet still be stable. mRNA therapeutics is perhaps the biggest challenge because you can modify it a little bit, but if you modify it too much, it doesn't translate. And for therapeutic editing, you can use oligos that activate the deaminase. And lastly, the work in Edigen and TrueCode are based on oligos that have to interact with the DNA repair machinery to work. So with that, I have the last slide, thanking the, hopefully got a lot. I'm sure I didn't hit everybody of the many people who have contributed over the last 40 years to the field. And I'll take questions. Thank you.
Great. Thank you very much, Todd. There is still time for some questions. So if people would like to unmute themselves and ask questions or else type them into the chat, I would be happy to ask the questions for you. Okay, so there is a question from Alison Baker. She asks, how does the off-target activity of the etomers compare to that of CRISPR?
Well, the interesting thing there is people are starting to use, I don't call them etomers, they're using modified DNA as donor DNA, modified oligos for CRISPR. So in a sense, when you're doing CRISPR, if you're doing a precise edit where you're changing one nucleotide with an oligo donor DNA, you have both the oligo and CRISPR. So we would hope it's less. But there's work done by Glaser. They looked at the specificity and that's published. But that was initial work. I think there's got to be a lot more work done to assess what the off-target effects are or whether indels are created. So far, they didn't see any, but I'm not sure they have looked hard enough.
I have a question, Frank, if it's okay.
Yes, go ahead.
Thanks, Todd, for this wonderful talk. You showed that mRNA, gives much more protein expression than the plasmid DNA, I guess, or the naked DNA that it was compared to. Do you know how it compares, and have they been able to improve how it compares to viral vectors, to AAVs, for instance, or adenoviruses in terms of yield, protein yield?
No, I would suspect that by the time you get a modern viral vector, it would work. You could get a lot more protein. The mRNA is going to be transient in a sense that it could last for a day or a few days or maybe a week or two, but it's not going to last as long as the viral vector. But I don't know the quantitative, how they've done it quantitatively. All I know is that when we looked at cell culture, back then we would take a plasmid and transfect it into primary cells in particular. It was notorious that they didn't express genes as well. I don't know if that's been resolved by some other method, but mRNA expressed very well.
Because as you know, the AAV, the issue with the AAV is sometimes delayed. I mean, it's often delayed expression. You need to wait like a week or so. So it's not to like avoid going for a viral vector, but maybe to combine modified RNA as well as AAV in order to get expression early on, even if it's not to the levels that you would achieve with a viral vector. Thank you.
Sure. Yeah, certainly the mRNA can come on quite quickly after transfection.
Kafir Azadmir asks, is pseudouridylation of mRNA enough to avoid innate immune system activation?
No, it is not. I mean, if you look at the oligos that have been working like for RNAi that are compatible with low side effects and not getting the sort of flu-like effects of oligos, they tend to have every nucleotide modified. And with mRNA, you just can't modify it as thoroughly. Maybe more recently, I might not be aware, but for many years, it was sort of explosive immune stimulation of the oligos, even when they were modified with things like pseudouridylation.
Great. We have a question from Anna Kraszewski. She asks, thanks for the great talk, Todd. What are some of the major obstacles that jeopardize the success of oligonucleotides as a principal class of broadly used drugs for various disorders beyond targeting the liver and the hematopoietic system?
Yeah, I think I did highlight some of the great moments where things were approved, but as Sudhir Agrawal pointed out to me this week, even with advanced chimeric oligos, I think he said 50 had failed in clinical trials. Even some of these splice blocking, people have gone at it, and some have worked and some haven't. So one thing is, you want to work with the experts like Alnylam or Ionis, people who know what they're doing, because you really have to optimize every aspect to get this to work as a drug. In terms of delivery to other tissues, that would be the limiting step, I think, outside of the liver. And that's being addressed by other ligands at Ionis. And the company I mentioned that Anastasia is working on is using her lipophilic chemistries, which goes into a number of cell types, as well as just the general phosphorothioate antisense goes into a number of cell types. If you could get over the kidney toxicity that you get with those, they may be able to extend that to many other cell types.
Thanks. We have a question from Feng Damon Wang. He asks, it seems like there's a big limit of chemical modifications in mRNA due to low yield of long oligosynthesis. Is there any ongoing effort to incorporate position-specific modifications in mRNAs like the RNA, siRNAs, and ASOs?
Yes, when I met with Moderna once, they asked me about how you might do that. There's certain ideas. I think in our original patent, we talked about ligating on a 3' end that might have modifications that you couldn't incorporate by transcription. If you're talking about, this is talking about mRNA therapeutics, right? So
mRNA,
yeah, so you could use RNA ligase and a splint oligo to put on an end or a 5' end that was synthetic and had multiple chemical modifications. I'm not aware of ways of making specific modifications in a very, very long molecule, but I'm sure somebody will come up with it.
Thanks. And we have another question from Fabrizio de Fajajana. And he asks, what's next for ASO? Which ASO features need to be improved with additional chemical modifications?
Well, it's, you really can do a lot with what we have. I mean, the affinity can be dialed up to, almost just ridiculously too high affinity, where, these LNAs are so high affinity, you don't want to use a lot of them because they'll get non-specific. You can make oligos stable for months in humans, a month or more. So a lot of the basic issues have been resolved by the existing chemistries, be they methylphosphonates or phosphorothioates and 2' modifications. I'd say it's complicated bringing it all together when you look at tox, delivery to the specific cell type, and combining all those with low immune stimulation. That's where it gets tricky. And I don't know if it's more different chemistries that we need or just different combinations of the existing chemistries.
Great. So there's another question about can you talk about the mechanism of self-delivery of sDRNAs?
Yeah. Well, it's interesting. When you make oligos that, let's say, are all 2'-O-methyl, that's where the first modified siRNA was. And if you did like a single or double-stranded all 2'-O-methyl and put some M-blocking groups on it, not phosphorothioates, it's stable and it gets secreted from the kidney instantly. So they're not that useful unless you put them in liposomes. And if you do that in cell culture too, I don't think those get into cells very well. When you start adding the phosphorothioates or a lipophilic group, as Mano showed years ago with lipophilic groups, and Rick Wagner also showed that, you start getting the self-delivering to occur. I don't know the mechanism. I used to laugh at Stan Crook, who's done all this great work, because one time at a meeting, someone asked him that question: what is the mechanism of your phosphorothioate oligos getting into cells? And he said it was a protein shuttling mechanism. And I looked it up on the internet. There was not a single reference to that word on the whole internet. But the idea I think he was getting at, and he's obviously a tremendous scientist who is responsible for developing much of the field, is that these things just stick to everything, the phosphorothioate tails and phosphorothioate groups, and they stick to proteins. And the idea is they just stick to the proteins as normal endogenous proteins are getting endocytosed, and they go along for the ride. But I'm not sure if the exact mechanism has been found out.
Great. Next question from Charles Jennings. Can you comment on getting oligos across the blood-brain barrier for CNS applications?
Oh, wow. That would be good. And it's even more important with the fantastic results that they've had with the oligos in the central nervous system. Yeah, I think the only thing I could think of with that's not involving something really crazy is the idea that they're working on making antibodies that go through the blood-brain barrier, and they would bind to transferrin or something and ride in. And they're also conjugating antibodies to oligos to antibodies. So if the people trying to deliver antibodies to the CNS succeed, then perhaps attaching the oligo, it could go along for the ride. Anyways, great to hear from you, Dr. Jennings. He's the Charles Jennings who saw his work on the specificity of oligos early in the talk.
Great. I think maybe the last question comes from Alan Herbert. He says that at last week's RNA editing summit in Boston, Wave reported the use of neutral backbone oligos in NHP producing editing of alpha antitrypsin that lasted for 45 days after a single subcutaneous injection, which was an exciting advance. So you have GalNAc targeting to the liver. Are there strategies to target other tissues?
Okay, so that example was GalNAc?
No. Could be GalNAc goes for alpha antitrypsin, but I think he's just asking what are the strategies for other tissues?
So other tissues, yeah. There's some work from Brett Monia, who's now the CEO of Ionis, and he went back and looked at some of their cancer work and their early delivery work with oligos that didn't have delivery ligands. And they do very sophisticated work now with measuring the uptake. And they were able to get a ligand to the beta cells in the pancreas and get delivery to those cells. That was a peptide that they conjugated to the oligo. So I would look up that work. But basically, it's not just GalNAc. That's a really great example. But there's always endocytosis coming, and you can ride in on these other pathway, other receptors.
All right. Thank you very much, Todd. It's been great to have you as our speaker. And thanks to everybody for hanging around.